All right, so today we are going to be going through my paper. There I am. So this here is 5 years of work, and this was essentially my thesis paper. This is Layilin inhibits integrin activation, and its loss results in platelet hyperactivation via RAC1 in inflammatory bowel disease. Don't worry, that's a lot, but we will be able to go through, and hopefully at the end of this, you will know exactly what all of those mean. Before I continue, please help me out by either joining my Patreon or dropping some cash via the Ko-fi link that I have in the description. Thank you! And now with the obligatory money talk out of the way, let's get back to the paper. Let's have the quick summary, shall we?
Paper summary as told by Science Goblin. Platelet bind to HA. Boop. What happened when delete?
Platelet go. If Science Goblin is my legacy on the internet, I'm totally fine with that. Now let's dive into what you need to know for this paper. Paper background as told by a boring lady with a PhD. So we start out with our favorite extracellular matrix, which you can think of as just a huge forest of proteins and glycocarbons on the outside of the cells that act as signaling molecules. We're going to be focusing on this one today, hyaluronan, which I've worked on previously in another video. So this is also called HA. If you're wondering what it's being attached to, that is CD44. So we know that during inflammation, there will be signals that will be sent that will cause a change in HA. What will happen is that HA will start to branch out into the bloodstream. These are the heavy chains that'll be used for multiple inflammatory processes. So we know that blood cells, like leukocytes, can bind onto them that can act as landing sites for further infiltration, like we see here. And what was important for my research is that platelets can land on and form clots or thrombi on these heavy chains. Here's the thing. This is what's supposed to happen when you're sick, but when you're healthy, you don't want these chains. So how do we get rid of them? Well, there are many ways to degrade HA, but we focused on the platelet functions to degrade HA. So there is an enzyme on the surface called hyaluronidase, hyaluronidase 2, and what we know is that platelets can then bind to and then start cleaving and cutting the branches to make these HA fragments. Now, quick note about these HA fragments as they're being formed here. 1, platelets are not the only ones who make these. 2, they're also known to be pro-inflammatory. We know that they can signal TLR4 in blood cells, and also known for endothelial barrier dysfunction. In IBD, we know that this platelet-mediated metabolism is lost and is reduced, which actually correlates to worsened disease symptoms. So a big question was, what's driving that loss? What's driving that dysregulation? In order to answer the broader question, we had to answer this specific question: what receptor recognizes HA? Hyal2 cleaves, but it doesn't recognize HA. This will be answered in figure 2 of this paper. Now, before I can get into what is Layilin doing and all of the data on Layilin that we collected, I first have to talk about a very specific pathway that will be crucial for this paper, and that is this integrin activation pathway. This here, on the surface of the platelet, is a receptor called PAR1. When an enzyme called thrombin interacts with that end right there, we have a cleavage of this site which will allow PAR to interact with itself. This will then lead to the phosphorylation of AKT, which will then lead to some more downstream signaling, resulting in something called a calcium release in the cell. This will then cause the activation of something called a GTPase. GTPases are the cell's natural on and off switches. When it's off, it's bound to GDP, and when it's on, it switches out into GTP. So this is its on form. Once the GTPase is on, it then results in the activation of PAC. which then results in phosphorylation, which can then interact with alpha-2b beta-3. This then causes a conformation change in the integrin, which will allow it to bind to fibrinogen. This whole pathway right here is called platelet activation. Now, there is another way for us to activate this pathway, and that is through the stimulation of GP6 with something called collagen. You will see me use that throughout the talk, but ultimately, we'll be looking at how Layilin interacts with the PAR signaling cascade shown here. And now for the paper figures as described by the PhD lady without a cold. So, here's figure 1. These here, in figure 1a, are endothelial cells. All right. In green, we have hyaluronin, which remember is that huge sugar that's on the outside of cells. And as we can see here in unstimulated, right here, this is no sick signals. We can see that the HA, or the hyaluronin, is just on the outside of the cells. That is what it looks like when you are healthy. And then if you're asking what the rest of these are, CD41 is looking at platelets. So anywhere you see red on here, like here or here, that's where platelets are binding. And then DAPI stains the nucleus. This helps us find cells. So anywhere you see blue here, well, that helps us find the location of that cell here, as well as here, etc., etc. So usually we use DAPI to help us actually find where cells are, and otherwise, if we didn't have that, we would just be looking at colors and we would not necessarily be sure if it was staining on the media that we had or staining on the plate that we had, or if we're actually staining the cells. So DAPI is actually a very important control and cell locator. Let me erase these really quick so that I can continue writing on them. So here we are exposing our cells to TNF-alpha, and which is a sick signal. This is an inflammatory signal. All right. So this is what would happen in your body when you are sick. Your cells will be exposed to TNF-alpha. And then what we see here is this unique branching from the endothelial cells. These are the HCHA cables that we were talking about. So this is the cables, and you can see on here in red, all of those little dots on there, those are platelets binding. And then here, in this last one, we have again the same sick and inflammation, but this is a control panel because we have exposed it to hyaluronidases. Hyaluronidase cuts up HA. So what we have here, you don't see a lot of green. So we low green, and that's because this is, again, a good control. If there's hyaluronidases, it's going to cleave the HA. Really, that's just proving that what we're staining in green is HA. That sometimes you look at those and think, why do we have these things? Well, controls are just there to go, what we are seeing is actually what we're seeing. So this whole A section is just essentially platelets. Whoa, that's not how you spell platelets. 1 second. Bind to HA. That's what this is showing. Let's move on to B. So B here is, using flow cytometry. You'll be seeing these throughout the paper, so let's walk through these. Here we have the agonist that we're stimulating with. An agonist just means the molecule that will activate the platelets. So here we have PAR1. PAR1 peptide, that's what the AP stands for, is a synthetic peptide. And this will stimulate our PAR receptor.
FSCA is how we measure size in flow cytometry.
But down here on the x-axis, this is measuring PAC1, which is active integrin. When we expose the platelets to no HA, we have 60% activation, about. However, when we expose it to HA, what we have is a decrease in that activation, about 50% reduction. So that's really interesting. So first A was showing us that platelets interact with and bind to HA, but now we're showing that HA binding to our platelets actually changes their ability to activate. Specifically, it is inhibiting. Let's go to C. So C here is again measuring PAC1. PAC1 is again measuring active integrin, so the higher this is, the more active the platelets that we're measuring. This is essentially just quantifying what B is. Okay, I know I spelled quantifying wrong. Forgive me. Sometimes I'm doing this off the top of my head, and we just accept that I can't spell. All right, so we have here is just HA, which is HA does not stimulate. That's what we're seeing in this lower bottom part here. And again, for simplicity's sake and for cleanliness, we're just going to erase some of that because there's a lot of red ink on here already. Sweet. So this first one, HA does not inhibit or does not activate. That's important. This is going to control. And then this is a just stimulating with our PAR peptide. And then we actually stimulate with an HA control here in green. So this is essentially cut up HA, which are not HA fragments. We'll discuss some HA fragments later, but in this case, it's such tiny HA that it's just bits digested HA essentially. And that also has no effect between the ability to activate. However, what we have here are these next two, which is the 10 kilodalton fragments versus the 500 kilodalton fragments. And when we expose our platelets to both of these, we incubate them and then we let them activate, we actually see significant inhibition of their ability to activate, specifically when we are, again, measuring PAC1. So B and C are showing the same thing. C is just showing all of the data together. All right. I'm actually, again, going to erase some of this just so that we have some space on here. Now D is looking at light transmission aggregometry. So this is what we were previously talking about in the experiments. What we are measuring here is the maximum aggregation. What you need to know, so we are measuring max aggregation, and the lower this line is, the more the platelets are aggregating. So here we are, again, stimulating it with another PAR peptide. You'll see us use different amounts of this PAR peptide throughout the paper. And that's just because for every experiment, it does change how much of the agonist you need in order for it to work. That's just unfortunately the nature of some of the experiments. But as we're activating this PAR peptide, as we're activating with this PAR peptide, we see, and when we don't incubate with anything, it has this nice curve right here. It really aggregates. That is a classic trace right there. However, when we incubate with two different sizes of HA, and again, we're using 10 kilodaltons versus 500 kilodaltons, both of them have very, 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 very similar aggregation curves. You'll see them start to aggregate right here, but then they go back, they stop aggregating. So this is, again, a significant inhibition of platelet aggregation. I'm going to just erase that for cleanliness. And E is quantifying all of that. So if we look here, we have all of our no HA incubation, and then over here we have our 2 HA incubations. And what I would like to point out here is that we do have statistical significance for the 10 HA. Here we almost have significance, and the reason why it's not the 0.05 that we want, it has to do with some of the math behind it, but this is still incredibly close, and honestly, many scientists, myself included, would just consider this to be pretty significant in mathematically. Sometimes you just have a wider spread, and that's what gives it that slight non-significance. But most people would consider this actually relevant to your claim, because the claim that we are ultimately making here is that HA inhibits platelet activation and aggregation. That's what this whole figure is showing. So here's figure 2. If figure 1 was showing how HA inhibits, now this one is asking what is HA binding to? So the first thing we did in order to answer what is HA binding to is A, which is a Kumasi stain.
For the sake of brevity, we can describe a Kumasi stain as running the platelet proteins through filtering proteins or glycoproteins that allow us, after multiple washes, to identify binding receptors. In the yellow box, you can see the band that helped us identify the HA receptor Layilin in platelets.
That was to help us identify even a protein that bound to HA. You're like, okay, where is it in platelets? And that's what B is answering. So this here is a sucrose gradient. So what you do is that you take your proteins. So again, we're going to have proteins here. I think I'm just going to keep to this thinner pen for this one. And then you actually can then take it into another tube, and then you can let the proteins settle based on their density. So fraction 1 versus fraction, for this one it goes to 10. Forgive me if you can't read that, but hopefully you're listening in on this. And so the proteins that are surface of the cell will be in fraction 1. And then fraction 10 will include protein in center of cell. I'm oversimplifying it, but you get the idea. Based on where the protein lands, we can then identify where it is in the cell. And then to clean everything up, I'm going to just quickly erase all of this. So what we're looking here, we're going to be focusing on these two. This here is cell surface proteins. So we have a well-known marker for platelets right here. This is CD42B. it should be on the surface for all of our platelets. This is our control. And then we have P-selectin. And as expected, P-selectin is right here. It's in 6 and 7. This is for the alpha granules. I didn't talk about them a ton, but these are the prepackaged granules. When your platelets are activated, P-selectin will be brought to the surface. But when they're just sitting around, P-selectin should not be at the platelet surface. All right, and then we're looking for our HIL-2, which is our enzyme on the surface of platelets that allows us to cleave. And as expected, they are, again, in the alpha granules. This is going great, which means our protein samples are, fractionating correctly. Things are going well. And finally, we can ask, where is Layilin? And it's actually in the cell surface fraction lanes. And then we have a little bit maybe in here on the alpha granules. It's debatable if there's like a ton of them in there. But we see that it is on the cell surface. So this is great. So platelet is already sitting out on the top of platelets. It does not need to be recruited in order for it to work. So then we go to C. Is Layilin the primary HA receptor on platelets? I know how to spell these. Jeez, it's nice, weird. So what we are doing here is a binding assay. So first, we take our platelets, and then we are going to expose them to blocking antibodies. So these antibodies will be binding to the surface of your cells. Let me just change. And then if we have appropriately blocked our HA binding receptor, it will also block HA from binding. No bind. So this is, again, if we have targeted the correct receptor, this will tell us that it should be the primary HA receptor. So first we have an isotype control. That is just, it should be a non-specific binding. Sometimes if you put antibodies in an assay, it will mess up the assay. And so this is just showing antibody won't screw up assay. Sweet. And it didn't. The binding is working correctly. Because what FITC HA is it is green, it is fluorescently labeled HA, and if it is binding to our platelets, it should have essentially 100% binding. This is actually fraction. The percent got in there. This is fraction. That was supposed to be fixed, but the joys of knowing where the mistakes are in your paper. All right, and then we targeted CD44, which is a well-known, well-studied HA receptor. It's actually debatable if it is on the surface of platelets, but to cross our, dot our I's, cross our T's, we still did anti-CDC44, and lo and behold, it does not change the HA's ability to bind. However, when we targeted Layilin, right here, we see a reduction of the HA's ability to bind. That means we are targeting the correct receptor. And then, this is in mice. Same experiment. However, this is Layilin knockout. We don't have Layilin. And this is where Layilin is present. And again, when we try to have that binding of HA right here, it works. And then when we have no HA, Layilin, no HA binding. Well, I'm saying no. Reduced. If I say no, it would mean 100%. none, but there's always some degree of binding. So you're going to say reduced. Fantastic. Now we're going to go on some more flow cytometry. And again, these are in human. This is again the PAR peptide. We're measuring active integrin. So the more it is over to the right, the more active it is. Look at this. This is looking very familiar. We again have, no antibody, and then we have about 60% stimulation or activation. And then when we block with Layilin, it's not actually blocking. When we bind with antibody, we have a reduction of activation again. And that here is quantified in F. We have this is just no stimulation, period. But here we have no treatment. That's what NT stands for. And then we have, again, our isotype antibody. The antibody doesn't screw anything up. And then when we let the incubation with this anti-Layilin antibody, we have a significant reduction. We have the star. It's not quite as intense as the HA molecule incubation that we had earlier, but this will become important later. I'll talk about it at the end of this figure. And then we have another aggregation assay over here. So G is aggregation. And again, the lower that line, the more it's aggregating. And in the presence of this Layilin antibody, when we stimulate with this PAR peptide, and over here with H, we see that, again, when we're measuring maximum aggregation from G, we put that all together, and we actually see that the presence of that antibody reduces the platelet's ability to aggregate. Now, pause here. This might seem counterintuitive because in the previous experiments, in C for specifically, antibody is blocking. So this is acting as a blocking antibody, but not in any of the following assays. In E through H, antibody is functioning as HA. And I can't show it because it's in supplemental data, but we did a lot of other experiments to show that the antibody for the rest of the experiments that we had, again, through E through H, it is effectively functioning as HA.
And so to summarize this figure, Layilin is the primary HA receptor on platelets.
All right, so this is figure 3. I'm only going to show, there's going to be some movement on this, so apologize. I'll try to edit out as much as I can. So now we're asking, where is Layilin in terms of proximity? And we'll be asking, what happens when Layilin gone? So this figure is kind of doing multiple things, but the first two is asking, where is Layilin in terms of proximity? So these are spread platelets. These platelets are on fibrinogen. So that is what they are spreading on. And what we have here in green is staining that alpha-2b beta-3, specifically the beta-3 subunit. And anywhere you see green, that is where that subunit is. And then anywhere you see violet in that same image, that is where we have Layilin. And then when you merge them together, anywhere you see white, that's where there's overlap, and that is where they are together. So what we're arguing here is that our Layilin antibody on the surface of the cell is also right next to the integrin antibody. Now, there is a slight problem with just showing that. What we had to show was B. Because from A, that's actually not an impressive figure if you know anything about platelets. Alpha 2B beta 3 is the most abundant protein on a platelet surface. So it would not be surprising, just because of how much of it is there, that you're going to have that overlap in a microscope image. If you were someone who's a platelet person, you would roll your eyes at if we had just shown this. So that's why we're showing B. This is a proximity ligation assay. And what it's doing is that we are labeling Layilin in the beta 3 subunit. And so we are labeling them down here on the surface of the cell. And then we tag them with antibodies that are specific to both. If those antibodies are close together, they will emit a signal. And that's what we're showing here, is that if we label Layilin and then beta 3, we have this shift in green. So the green shift here, this little curve, is a positive result for this signal. So this is showing that this Layilin and beta 3 equals close. And we also did some things in the supplemental data. There's a really cool pull down assay that I did. Can't show it, but it was also like a third proof that Layilin is in proximity of alpha 2B beta 3. For cleanliness, we only showed the first two, but there is a whole another figure in the supplements that helped with our claim here. So let me move this just a little bit so we can see this better. So now that we have shown that Layilin is next to alpha 2B beta 3, the next thing we had to ask was what happens when it's gone? Does it affect alpha 2B beta 3 signaling? So what we're doing here is, again, we are stimulating PAR with this PAR peptide. And we're measuring in whole blood. Whole blood is important because it is where the platelets have been least processed. They have been least jostled around when you're isolating them. So doing whole blood experiments is incredibly valuable. You just can't do much else with whole blood in our lab. But what we are doing here is that we are measuring Ajanel A. And this is all in mice. So anywhere you see gray is a wild type mice. Anywhere you see red, these are the mice that we have completely deleted the Layilin out of every cell in their body. And so in red are platelets that should not have Layilin. And what's exciting here is that we see the red outcompete the wild type here. I'm saying outcompete, but we see that the red is more active in terms of the Ajanel A activation compared to the wild type. Red equals hyperactive. So this is a hyperactive phenotype. And then we measure P-selectin. P-selectin is, again, a molecule that when platelets are activated, it can be brought to the surface, which will allow the platelets to stick to other cells. And there's a little bit of significance here. Oh, gosh. See, the thing is that, you notice small mistakes. Oh, I don't know how those bars disappeared. But this here is the only little bit of mathematical significance that we found. Otherwise, between the two, when you stimulate with PAR, not as big of a difference between the wild type and Layilin knockout. Let me move this. Now we are doing that GP6 stimulation with convulsin. Convulsin is essentially snake venom. It's really cool. And again, we're doing it in whole blood, measuring Ajanel A activation and P-selectin activation. And there is a little bit at the lower concentrations of convulsin. We do see that hyperactive phenotype in our knockout. And we actually do see more of a phenotype in our P-selectin over here, but it's not nearly as pronounced as our Ajanel A when we stimulate with the PAR receptor. So now we want to then stimulate with thrombin, because previously we've been doing everything with this PAR4 peptide, but your body does not make the PAR4 peptide, your body makes thrombin. Well, in order to use thrombin, we need washed platelets. So we are still simulating PAR, but now we've jostled the platelets a little bit more as we've had to wash them a bit, and then we have to let them sit and whatnot. So these have been processed a bit more, but you'll be excited to see that thrombin still has this hyperactive phenotype as we increase the thrombin concentration. And then even with P-selectin, we see a little bit of a hyperactive phenotype there. Not quite as pronounced, but you do see a bit more than from, just whole blood. And I'm going to move this real quick. And now we get into GP6 stimulation with the same washed platelets. And unfortunately, again, we don't see much difference here when we look at the Ajanel A activation. So there is a P-selectin phenotype in our convulsin. However, because we have such a huge difference in our phenotypes and in our between our wild type and knockout when we measured with thrombin and with the PAR peptide for Ajanel A, which is like up here, I'm going to circle it on here. Because that was such a huge difference, we decided to continue pursuing that. Like, that's where we thought the money is. I still think that there is something to be seen here. Like, there might be something with the alpha granules that we can look at for the convulsin. But I did not pursue that when I was doing my PhD. So now we have figure 4. Figure 4 is, again, this another aggregation assay, that light transmission aggregometry. And again, anywhere you see this curve go down, the lower the curve, the more it's aggregating.
What we see here in A is the trace that we have of the aggregation curve, where we have wild type going lower or clotting more than the Layilin knockout, which is going to be quantified in D. And again, this is when we're stimulating with thrombin. Despite Layilin knockout being hyperactive, clots were unstable. Though the traces may not look impressive, that is a 20% difference that we have between those two traces. Just a reminder, as we're looking at the impressive quantification in D, gray is wild type and red is Layilin knockout. Look at that significance. PAR1 is a G-coupled protein receptor, and so we decided to target more G-coupled protein receptors with arachidonic acid, which is what you're seeing in B and E. I write PAR here just to remind you it is also a G-coupled protein receptor. So the question is this unstable clot in our Layilin knockout for G-coupled protein receptors in general?
If you look down at E, when we're stimulating with this arachidonic acid, we can see, again, there's this reduction of Layilin here. When we stimulated with collagen, which is the, GP6, there's a G there, receptor, you don't see a difference.
What this is showing is that though when we stimulate the PAR receptor, we will have increased integrin activation, we also lose aggregation capabilities due to complete deletion of Layilin. That is, their clots are unstable. There's a whole other pathway that's been disrupted by the deletion of Layilin. This is certainly an area for future directions to look into.
But yeah. So, this is the venaflux figure. This is where we are actually seeing how our platelets bind under flow. And so we have venous flow, and venous is the slow flow. This is what you will have in your veins. Again, we have wild type up here, and we have our knockout down here. And what we do is that we look after 1 minute, 3 minute, and 5 minute of the platelet's ability to bind. And even at the first minute, we can see the starting difference, and then more and more. And then when we quantify it over on B, we can appreciate that the Layilin knockout in red has way more binding area than compared to the wild type here at both 3 and 5 minutes. This is happening on fibrinogen. Fibrinogen, if you'll remember, is what alpha-2b beta-3 bind to. So this is extremely exciting because this is still going off of Layilin knockout equals integrin hyperactivation. Moving this down. All right, now we tried to have it bind to collagen. Same thing, wild type Layilin knockout. And we just don't see a huge difference between our Layilin knockout and wild type.
Please head to part 2 to see the discussion on the figures.